Hello, everyone, and thanks so much for joining us today for the latest Technology Network's Ask Me Anything. My name is Lucy Lawrence, and I'm the Senior Digital Content Producer for Technology Networks, and I'm very excited to be the host of today's session. And I'm very, very pleased to introduce our speaker today, Professor Michael Shuler at Cornell University. As the name suggests, this AMA session provides you with the unique opportunity to ask our distinguished guest absolutely anything about their work, background research, science, and innovation in the field. But before we do begin, I'd like to highlight just a few important details. So, firstly, this is a live session, and we are thrilled to have so many participants joining us today already. Many of you have actually already submitted some questions when you registered, but don't worry if you haven't, because there is still time. You can add your questions during the live session. Simply type your question into the box that you'll see on the right-hand side of your screen and click send. We'll do our best to get through as many questions as possible before the session concludes in roughly about an hour's time. If you do run into any technical issues throughout the webinar, just click the chat icon in the bottom right corner of your screen to request support, and a member of our team will be sure to help you out. So, without further ado, it is my great pleasure to kick off this AMA with our first question of the day, Michael. Could you perhaps start by sharing a little bit about your background and the initial conception of Body on a Chip, and what were your motivations in doing it?
Okay, well, thank you. My background is trained as a chemical engineer, but I trained really as a biochemical engineer, which was a little bit unusual back when I got my bachelor's degree in '69 and my PhD in '73. Then I started at Cornell in 1974, and I worked on a variety of problems involving biochemical engineering. And I'm also currently now president of Hespros, a company that's commercializing body and chip systems. So in terms of a kind of background, I was, in terms of this particular idea, I was in SPAC at the University of Wisconsin in '88, '89, and I wrote myself a letter, postmarked June 12, 1989, and that's established this idea in terms of for patent applications, basically. The idea of building what we call a cell culture analog, or artificial animal, artificial human, to use in toxicological studies, or perhaps more importantly, in preclinical drug trials. We thought multi-organ human-based surrogates could lead to better choices in terms of drugs to take into clinical trials, and also allow testing of drugs on various subpopulations of humans. The last issue is when there was a personal motivation for me. Our oldest daughter has Down syndrome, trisomy 21 and 3 copies of the 21st chromosome. And we found physicians can sometimes be reluctant to prescribe even FDA-approved drugs for people with Down syndrome because they had not really been tested on subpopulations like that. So part of what we put into this original disclosure was that we could build not only human-based models, but human-based models of different subtypes. So, and Down syndrome was one example of that. So that was a basis. And again, many drugs in clinical trials fail not because they don't work on the majority of the population, but there's some adverse reaction with some subpopulations. So being able to build subpopulations potentially is an important possibility. Overall, animal models do a fairly poor job of predicting human response to drugs. So I know, like at least one large pharmaceutical firm I know, 5% of the animal studies in preclinical trials turn out to be predictive. Overall, the statistic is 11% of drug candidates that pass preclinical testing using animals, enter clinical trials, and exit as approved and useful drugs. That means that 89% don't. So a human-based surrogate, I think, could be a missing link to improve that. Clinical studies are very expensive. Mm-hmm. And the capacity to do such trials is limited, so it's important for society to choose the best candidates to go into clinical trials. Because every time you put a loser in there, it means that another compound that could have maybe succeeded didn't get to make it in there. So making that choice is important. And we're not going to make a perfect replica of humans, but if we could change the success rate from 11% to 33%, that would have a tremendous impact. That would be 3 times as many useful drugs for the same investment, basically. Um, and again, if we have human-based analogs available, they'll certainly lessen the need for animals in studies, potentially make better predictions of human response, and both in terms of drug efficacy and also safety by identifying potential side reactions. That's why we're interested in multi-organ systems as well, because that gives you the possibility of looking at not only the response in the organ of interest, but also toxic side reactions. So we asked, how can we build a human surrogate for drug testing? We wanted a system that connected various organs and cells in a physiologically realistic manner, not simply a multi-organ model, but one that was physiologically realistic with a recirculating blood surrogate. So a tool developed by chemical engineers, and my training was in chemical engineering. Widely used in pharma is PBK models, or physiologically-based pharmacokinetic models. You divide the body up, animal or human, up into different compartments. They're interconnected like they are in the body. And the relative size of the compartment must be physiologic. In other words, you want the liver to be larger than the pancreas rather than vice versa. Within the model organ, there should be appropriate cell types and diffusional distances recognized, etc. If you're going to have a good model. And again, we might build a model of an organ to have 40 cell types. We don't probably put all 40 cell types in. We may put 2 or 3 most important ones in there, but enough to try and capture the important biological complexity in the organ. The blood flow rates have to be are designed to get, well, if you have chemical reaction engineering theory, you realize that the degree of reaction in a reactor does not depend on the size of the reactor. It depends on what's called the residence time, which is the volume of the reactor divided by flow rate. So whether the reactor is 10,000 liters or 10 microliters, you get the same degree of conversion of the substrate, the same products formed. And so as a consequence, if we were going to build these microsystems and want to have them represent humans, it's important that because we don't have to build something the size of a human, we can build something much, much smaller. And that's critical, I think. And originally when we did these systems, we did them at a macro scale, little beakers and stuff like that. But by 1998, I started working with Greg Baxter at Cornell Biotechnology Center, and we constructed microscale devices. The idea of scaling, 1 to 50,000 to 1 to 100,000, depending on the question, it might be a little bit different. But by going to that small scale, you can build a body and chip with relatively low cell material. And cell materials can be the most expensive part of a lot of these things. So you get, and you can build many copies to run in parallel. So that gives you much better statistics. So going to the microscale really makes the potentially economic to do this in a joint development framework.
No, fantastic. Thank you ever so much for kind of sharing your background and a little bit about your story as well. And you did mention you founded a company. I wondered if you could maybe tell us about the history of founding the company and why you think a human surrogate is important in the context of that too.
Okay. So I've been working with Jay Hickman down at the University of Central Florida for a few years. And in 2014, we decided that we would form a company to commercialize the body and chip concept. We announced in a meeting in November 2014. Our first employees were added in 2015. We founded the company as a CRO, contract research organization. So we decided not to sell devices, but rather companies would come to us with their problems, their compounds, etc., and we could provide a platform to do the testing for them as they wished. And because the technology is still fairly complicated, and as a consequence, we were concerned that people who were not that familiar with the technology might have difficulty getting it to work, and if you
Mhm.
didn't get it to work, would say, hey, this isn't worth anything. So we wanted to keep it in-house that way. So we've done it pretty much always on as a CRO.
Mhm.
Uh, and there are a lot of CROs that are very successful, you know, animal-based CROs, etc. Um, so it's, I think, a viable option. Now, again, we worked, our work here at Cornell was largely on multi-organ systems, but where we're principally interested in metabolism and following conversion of compounds and things like that. Jay added an important another dimension to it, and that is he works with electrically and mechanically active tissue, such as cardiac and neuronal and such tissue combined with uh, microfabrication. And that's critical in the sense that if I'm looking for the formation of a compound in circulation medium, that can take a long time.
Mhm.
If in fact, let's say it's a cardiac system, the response is in terms of minutes or less. So having that cardiac tissue there allows us to look at a drug or a compound that may modulate the activity and see if it, for example, does modulate the cardiac response. And that's much more informative than having to worry about cell death as a marker, which would be a long time away. And also, if it is a cardiac problem, we can determine whether it's electrical or mechanical. So start getting at something in the mechanism at which there's a problem. So, so Jay has spent his career working principally on a lot of these electrically active tissues, particularly neuronal systems. A lot of our success has been with neuromuscular junctions and things like that. And so it was a good meaning of the minds to combine his approach with our approach, and it enhances both of them. Uh, so that was, when we formed the company, that was what we had in mind.
Mhm.
The Again, in the last 10 years, we've grown from uh, you know a single employee in 2015 to over 50 employees now.
Mhm.
And we operate again with a combination of grants from the government, which is kind of our R&D.
Mhm.
But the government pays for it basically, and we've been fairly successful in getting grant money. And then we work with contracts from commercial firms.
Mhm.
Commercial firms providing, this is the problem we have. Can we solve it? Can we help them with that? And sometimes I have to go back and forth and say, we can do this, maybe not that, but what do you need? And work that out. And uh, we started off working with a variety of large companies. We had some papers, with Roche and
Mhm.
AstraZeneca, et cetera. Um, but we found that actually for this technology right now, most of our customers are smaller to medium-sized firms.
Okay.
Larger companies can kind of afford to do this on their own, and they don't necessarily have to come to us. But for the smaller, medium-sized firms, we offer this as a service that they really can't put together on their own.
Mhm.
Um, and we've had a fair amount of success with rare diseases.
Okay.
Um, uh, there, where in particular you can repurpose a drug, so we can measure potentially efficacy and toxicity. But if we have
Right.
a drug that's already been approved for safety, we don't have to worry about doing toxicology.
Mhm.
We have to just show that it works, and that is for many of our customers very attractive because um, it speeds things up, it reduces cost, and it helps them solve a problem. And there's like 7,000 rare diseases, so there's no end to the possibilities.
Mhm.
So it's been something we worked with. We do work with, we're happy to work with cancer, we're happy to work with heart
Mhm.
disease, but I think some places where this technology has had maybe the bigger impact is with rare diseases, where often there
Yeah.
are animal models. There's no animal models to
Yeah,
reach.
of course. Mhm.
So that's kind of where we're at right now.
Mhm.
And
Yeah, that's nice. I imagine it's had quite an impact on clinical trials, for example?
well, we are seeing an impact, and again, that's showing that the technology is maturing
Mhm.
and being accepted. The First case involved a potential treatment for neurological disease such as MM. Uh, and our results were enable a filing of a IND investigational new drug test uh, with FDA
Hmm.
and allowed the company, which was Sanofi in this case. It was actually a smaller company we worked with, and Sanofi bought them out and then
Okay.
taken clinical trials. Um, that started in April 2021, so it's been a while ago that it
Yeah.
did.
Hmm.
Phase 2 was successful and
Hmm.
now proceeded to a phase 3 two-arm clinical trial in 2024. So
Nice.
that is, um, that's very encouraging to us. Again, it's going to take a while. Again, hopefully this will be successful and it'll be approved as a drug. But again, you just have to wait to go through all the data, etc.
Hmm.
But and in this particular case, this was again an example of a rare disease. The drug that was used was a repurposed drug that had been shown to be safe, but now it could be repurposed for this use.
Okay.
So that was important. Now
Hmm.
we also have some other INDs that have been and clinical trials, phase two, rest are phase two clinical trials.
Okay. Hmm.
They started more recently. Uh, uh, Dianthus for MG, myasthenia gravis is again another neurological disease.
Hmm.
Uh, Agenix for ALS, which
Yeah.
has a clinical trial going. There's an often drug designation for ALS with Apellis, and then we just have another IND that we're working with Vanda Pharmaceuticals and getting established. So, you know, we've at least shown that the technology can have clinical impact.
Hmm.
And again, we're very interested in expanding this. We're working with a large variety of companies. So, and again, not all of them I can mention,
Hmm.
but there is significant interest out there, and it's so I'm very hopeful that the technology as a whole will
Yeah.
mature and be useful.
It sounds like it's having a real impact. So, what do you kind of see in the development of MPS technology in the next, say, 5 years or so?
Thank you. Well, again, there are different forms of MPS.
Hmm.
A lot of people, at one point, there was like 65 companies. I'm not sure how many are left right now. Um, but most were in the mode of more organ on a chip, single organs.
Hmm.
And that is a technology which, it can be very important in terms of developing certain drugs. It
Hmm.
gives you less overall information, but maybe very critical information on a human response for a particular application. Um, so I think that will continue to be important, and that will then be incorporated within a more overall approach. Um, in our case, and there's about 4 or 5 other companies like us that are more interested in maybe the whole system as a whole, and and I think there's going to be again a lot of use for that.
Hmm.
I think for what we're doing, initially we're going to see the rare diseases are the easiest because again you just have to show efficacy, not both efficacy and
Hmm.
the toxicity if you're working with a repurposed drug. So that's easier to demonstrate for FDA. So I think a lot of that will happen. I think you know in 5-10 years, um, the human base body will start to replace animals.
Hmm.
I mean, animals still you still may want some animals, but the human-based will be far reduced, and human-based models are probably very important. Again, like I said, there are over 7,000 rare diseases, so I think.
Yeah. Hmm.
Um, and in general, we would expect MPS to maybe largely replace animals, at least for efficacy testing. Um, Now, in terms of efficacy and toxicity, we expect that we will see these systems making significant inroads.
Hmm.
There'll be less so than for where you only need the efficacy. Um, but I think we'll see gains in both. And again, we have indications of that also, some of the other things we're working with.
Hmm.
Um, the Now, I mentioned this all in terms of drug development.
Hmm.
There's also another application for this, which
Okay.
is important, and that is chemical toxicity.
Yes. Hmm.
If I have chemicals, I could use this system as another test rather than going through animal tests to determine if it's going to be a potential problem for humans. And
Hmm.
one of the nice things about this kind of system is you can use it for multiple compounds at one time. You can test it with 3 or 4 drugs. Ask how do they interact with one another. We do that with drugs, but particularly for chemical toxicity, it's very important because sometimes the chemical by itself is not toxic, but interacting with other chemicals, you get a
It can
toxic.
be. Hmm.
So, and of course, that's extremely hard to test. So, you know, the market for doing that is not numerically as large in terms of dollars, but it's certainly another possibility for these systems. And I think one that would be very useful for society as a whole. And also, I think another thing again we haven't seen and we're not working on, but I can imagine a time where you're running a clinical trial, but you run an MPS along with the clinical trial. You
Mm-hmm.
do that in the terms of sometimes being able to better understand mechanism, and you do some tests on the MPS system while you're doing the clinical trial. I've
Hmm.
seen this. What does that mean? Can I use this to better understand what's happening? So I think that's another potential application that, again, is not near term, but it could happen in 5 or 10 years, something like that.
Yeah, and I think that could be pretty exciting. So in terms of, we've touched on those big developments that we're hoping to happen within the next 5 years. What do you see as being the biggest challenges in the MPS sort of body on a chip field right now?
Well, again, it's one thing is regulatory acceptance. We're making progress that way.
Hmm.
Um, but if for companies, particularly large companies, to adopt the technology, they're going to have to feel very comfortable that the regulatory process will be. And again, FDA has made some changes. Uh, they've said they're very open to body and chip technology. The Drug Modernization Act, FDA Modernization Act, actually requires them to be open to this kind of technology. But it takes a while for that to happen. Both regulators and, in this case, the drug makers have to be confident in the technology. So I think, again, we have these few cases now in clinical trials demonstrating it can work. The more cases like that, the more it's going to kind of snowball and people get it. But that, I think, is probably one of the biggest hesitations. And again, they should be. You want these drugs to be safe. You want to cure people. So you don't want to be adding something to somebody just as a whim.
Yeah.
Because you
Hmm.
think it works, you really need the data. But I think that's going to happen. I think FDA is open to it. FDA will help. They'll ask the right questions because they want to make sure this is going to work. And again, that's going to push developers to make sure they think about what they're doing. But I think that's going to be probably the biggest thing. The other thing that will help is uh, as people become more used to these systems, uh, and there are more and more of these systems used, the cost will come down. Right now, again, if I'm doing just the efficacy testing, uh, and you had to do animal testing before, we can be a lot cheaper than just doing the animal testing.
Hmm.
If you're doing animal testing, toxicity testing, then it's a little more difficult in terms of the cost proposition. We think we'd be better. We think, but that's a barrier that people always will look at is the cost of doing this and the quality of the data that you get. How useful is the information? If the information is really useful, uh, you will pay more for it because the animal data may not tell you a lot about how it's going to happen in humans. So you can, uh, but it's I think those are well, if a pharmaceutical manufacturer knows that the FDA and regulatory agencies are going to look favorably on the results if they provide them the right data, it's going to help MPS. And I think it will happen. Like I said, it takes several years from the start of a phase 2 clinical trial before you get an approved drug.
Hmm.
So just to have examples that it works will take a while for people. But once people see some of those happening and they gain confidence in the system, it'll be a snowball effect.
Yeah, I hope so. So we do have a couple of live questions that have
Sure.
come in whilst we've been having a little chat here. So one of the questions is asking, they are wondering if the technology could be leveraged for insect cell-based assays. Do you have any thoughts on that?
Uh, well, it probably could. I actually have done insect cell culture for production of pharmaceuticals.
Nice.
So there's no reason you can't. You could do this with any system. You could build an insect cell model. It's not one that we thought about. We can build a plant model if we wanted to. And again, those are all possibilities. Um, so it's possible. I haven't given much thought to it, but if I were looking for treatment of pesticides, I guess
Hmm.
you could imagine, and this is just imagining. Uh, you can even have an animal model with an insect as a portion of that model and see how it responds in that case. Um, we are interested in some systems right now where we're building models that have a gut microbiome in it. So you're building a system which has uh, another organism involved in it, so you can see how it responds, but also in the context of the larger organism.
Nice.
Uh, but with plants, I think you can do plants too. I did a lot of work in plant cell tissue culture early on, like the production of taxol.
Hmm.
Uh, so I would not be afraid to use it for those things.
Yeah.
Uh, Now, and again, part of the situation is the economic value. Who's willing to pay for it, how much are they willing to pay, make a difference of whether you would want to do it or not.
True. Hmm. No, perfect. Thank you very much. Another question has come in here from Lindsay, who says, "Thank you so much for sharing your expertise. Could you comment on the possible value of chips built with animal cells? Do you think it's worth exploring further?"
Oh yeah, no, um, and we do build sometimes models with chips with animal cells. Um, uh, so partially for veterinary medicine, you could see obviously the same thing. Although again, there it's partially to augment probably the studies you're doing because clinical studies and animal studies can be the same thing. Um, but there's no barriers to doing it. In fact, conceptually, we have built a lot of animals. We've built, for example, rat models to ask how could we take current rat data, understand that, and use that to extrapolate to humans. So building animal models with animal cells is really probably easier because the cells are often cheaper than it would be if than with humans. So humans is the bigger challenge. It's also where, again, I think most economic impact will be, but we can certainly do animal models very easily.
Hmm. Oh, perfect. Thank you ever so much. Another question that we've had in, so these are kind of dotting all over the shop, but this one is saying, just wondering if you construct MPS with less cells than they exist with in vitro, do you notice the differences in cell adhesions, or do you kind of factor in physiological cell superstructures?
So we do some things with superstructure. For
Hmm.
example, building a model of the colon, we can build it where we have the villi and the cells in appropriate locations. So we can take into account structure, at least at some level. Um, the um, Sometimes you even can see some self-assembly by cells will self-assemble in terms of structures which represent what might be in the body. Um,
Hmm.
so, in fact, it's kind of a trade-off between what we call engineered systems, where I put the cell right where I want it, etc., versus systems which allow them to self-assemble. So, organoid culture would be an example where
Yes.
it's self-assembled. Now, again, we have done systems where a portion of the cells we're looking at are in organoid culture. So we don't have anything against organoid culture. We make use of it where it makes sense. Now, sometimes it does make sense, sometimes it doesn't. You gain some advantages from an engineered system, sometimes in terms of control, but the self-assembled system you feel may be more confident in the structure that comes out. So there's trade-offs, and you know, this body and chip system works with either one.
Mhm.
Most of what we do are engineered systems because in terms of the measurements of output, a lot of times, we need the cells in a certain orientation to measure an output. But we still potentially could do it with a self-organized system. So
Mhm.
we have run some systems self-organized where I would say we generally tend to think of engineered systems, but we do use and can use organized, self-organized systems.
Nice, perfect. Well, thank you ever so much. So we're about halfway through the session now, and of course, this is a live session, so we do encourage you to keep those questions coming in. Another question that we have here that's lovely, actually. What are the major side effects of using chips in biomedical technology?
Okay, so the so I'm not quite sure what to mean by biomedical technology. In terms of
Mhm.
what we're using, the
Yeah.
chip is a host. And again, the issue is not just the chip, but how you modify the surface chemistry. So the surface chemistry is very important to the overall response here. So if I have just a silicon chip where I have not modified the surface chemistry appropriately, uh, then the response I'm going to see is not going to make sense. Again, I mentioned Jay Hickman's contribution in terms of the electrically active and mechanically active tissue. The other one is he's a surface chemist by training, so he has a lot of expertise in surface chemistry. So part of that is that when we build the chips, we have to worry about the surface chemistry we're going to put on them. When we design a medium that we're going to grow the cells in, we have to worry about how it interacts with the surface chemistry. What absorbs, what doesn't absorb, how can we make it so it comes out in solution what we want it. So that ability to do that is actually important. Uh, so now again, it wouldn't have to be a chip. I guess we could carve it out of wood or something like that and solve the same issue. Uh, but the microfabrication technology is very easy to use. Now we can do microfabrication with silicon, but you can also do it in a lot of different polymers. So, and sometimes that may be advantageous if they have a more softer tissue rather than hard tissue in terms of particularly mechanical deformation. So we build a lot of systems where we use, polymeric materials, sometimes soft polymers versus hard polymers. Uh, so again, it depends on the question you want to ask, how you want to modify the surface. And the question we want to ask is either one that we're interested in, want to sell to government for NIH for support, or we have a customer that has a particular issue they want, and then you have to think about what is the surface going to look like. Now, again, uh, I don't think we're going to go into the business of building beaker size things anymore. It'll be micro scale, but you can do a lot with microfabrication with soft materials. So there's a lot of expertise out there. So we can go to relatively soft materials without any problem. If in fact that's what makes sense in terms of the context of the question we want to
Mhm.
ask.
No, perfect. Thank you ever so much. Another lovely question here is, what are your thoughts on the future of a gut brain on a chip system? I know you've mentioned, you kind of touched on this. And how could it impact research in neurodegenerative or neuropsychiatric disorders? Because this person finds it fascinating and would love to do a PhD thesis on the subject. So really wanted to hear your thoughts.
Okay, so it's certainly possible. Um, we have written a proposal just recently, submitted in December, on looking at the gut-brain axis. So I can't say too much about it, but we do have gut models. We built a lot of models over the years of the gut, and so that's not a problem. This would be a gut model along with a microbial biota model because it's really the interaction of the microbes that are important, in particular some of the compounds that they make, which then are transferred through the peripheral circulation into the brain. Um, uh, Sometimes they act directly in the vagus nerve, but let's say circulate to the brain. We have a blood-brain barrier model. That's one of the things my group has worked on for a while, and
Hmm.
pretty good blood-brain barrier model. And then Jay has a model of the brain with the cortical neurons, the microglia, and the astrocytes.
Wow.
And with that, he can look at what's called long-term potentiation, um, which is kind of a surrogate for memory.
Okay.
It kind of you function as in the brain. So that is, you wouldn't necessarily think about, it, but you can actually see something about the function of the brain in response to, say, change in the microbiota in the gut. And so we think we have a model that can do that. Um, I don't know if the reviewers will agree or not. That's another issue. And but it's certainly something possible to conceive of doing.
Yeah, fantastic. Thank you ever so much. Um, Questions are kind of going to be all over the shop, but this is a really nice one. What do you think is the most exciting potential application of body on a chip technology that is still in its early stages?
Oh, um, well, from my point of view, almost any application is exciting.
Yeah.
The I think one of the things that's often very difficult to do. Let's say we're looking at animal models.
Hmm.
Replicating certain human diseases can be very, very difficult in animals. So the fact that we could represent certain human diseases in our model that really are not well represented in animal models is really, I think, a big plus. Now, again, uh, Alzheimer's is obviously one that comes to mind as I think
Hmm.
that we think we can say some things about with our model. Um, it's certainly an important problem because just economically uh, for the society as a whole, and as the number of people like myself get more aged, you have this potential problem looming out there, and the economic cost of it is extremely large. So having ways to look at potential drugs and do so in a way which you can screen maybe lots of possibilities, I think is exciting because uh, you know testing on patients and people is difficult. Endpoints are difficult. So now again, part of this depends on having some endpoints that you think you can use,
Hmm.
but there are some that we think we can use. So we're doing some work on with Alzheimer's and
Hmm.
this. Uh, so I think that would be one that would be exciting to do, one that it would be really impactful, and one where there are not too many other good alternatives.
Yeah, no, fantastic. I actually think that would be really, really exciting. Um, so let's have a look at another question that we've got here. Given the rise of sequencing technology and its use in personalized and precision medicine, do you also see the value of MPS technology for the purpose of personalized and precision medicine?
Yes. Uh, so the is one of the things we talked about. One of the things we thought about, and we can do it. We have done some very preliminary things with it. If in fact um, we have someone now, often we work with iPS cells, uh, but and we could get iPS cells derived from a certain particular patient and use it. Uh, we can use primary cells. Uh, Using primary cells sometimes presents more difficulty in terms of the cell culture aspects of it, the media that we use, and so it's that can be a little bit of a challenge. But I think that's we certainly can do it for personalized medicine. And again, when I talked about people with Down syndrome and subpopulation, that's really starting to get a personalized medicine, right? We're starting to ask a patient has a certain genotype, what is the right response? And I think again, from the point of view of drug development, I'd like to see a time, let's say there's drug companies who routinely run this through a population subtypes, representing people of different genetic background and people that they came from Asia and Africa and Scandinavia and places like that, and how they respond differently, and also again, are there genetic diseases? Again, I think we can do that fairly easily, a little bit more than personalized medicine. But we can do personalized medicine. We can build a chip for a particular person. I think that is the biggest expense there will actually be getting the cells of the person in a way that we can use them and adapt them to our system.
Mm-hmm. Thank you so much for your answer. We've had so many brilliant questions come in, and do remember that this is a live session, so do keep them coming in. Another great one here is asking how far away are we from the inclusion of the lymphatic system and immune system in sort of body on a chip platforms? And then secondary to that, what about brain organoids?
Okay. Uh, Brain organoids, potentially useful. We can build a blood-brain barrier. Uh, we have brain organoids around I guess, to a blood-brain barrier. I haven't thought much about it, but we can use organoids. In some cases, sometimes people have ways to take organoids and spread them out like that, so that they become 2D. I know Don Ingberg's talked about that with some intestinal systems. I mean, that would be fit into the system very easily. But real organoids we can use. It it's a little bit more challenging from the engineering point of view, I think. In some ways, it's simpler because you're not engineering the system. But in terms of how do you access the information you want, how can you observe what's happening inside, it becomes a little bit more difficult. So those are the trade-offs there. Uh, I lost the beginning of the question, though.
No, that's okay. Sorry, so it was kind of two-pronged. Let's have a look, see if I can find it again. So it was how far away are we from the inclusion of the lymphatic system and the immune system as well, from being
So
integrated into that?
we already have some papers we published where we have some aspects of the immune system already included. Uh, Now, not the full immune system, but some aspects, particularly with circulating monocytes and things like that, we can put in there and see how that responds. Uh, we're working towards that. Not a full immune system, and that would take a few years, but certainly aspects of the immune system we can certainly include and have included.
Nice. And then, sort of secondary to that, we've had another question come in saying, is it possible to have things like muscles on body on a chip systems?
Yes, yeah. So one of our standard ones, in fact, is a neuromuscular junction. And this is something that Jay has developed a long time ago. But basically, you can look at the response, the nerve connected to the muscle, and the response to things that might stimulate the nerve and how the muscle responds, vice versa, perhaps. We have also done other things where we built just muscle tissue into a system. Uh, that's certainly possible. Muscle is not um, uh, perhaps that difficult, but I think neuromuscular junctions particularly can be particularly informative for us. And a lot of the diseases, neurological, involve a neuromuscular junction, so that becomes a key target for us.
Nice. Another question we've had in is how sort of body on a chip systems could integrate within spatial AI technologies and about the potential of these systems in modeling neurodegenerative diseases like AD or PD. Do you have any thoughts on their applications in drug development there?
Yeah, so I'm probably not the best one to ask about that. I don't know very much about AI. I mean, I've heard about it, read about it, but I don't really know much about it technically. Um, I would imagine the systems we have you can analyze very closely. One could analyze in a way in which you got numerical and quantitative information, which you could probably put into an AI model to make some predictions. But I really don't have a lot of expertise to add to that. I think it's possible, not anything that I've thought much about. And it's Because one of these technologies, I think we're going to see how it sorts themselves out too.
It's still fairly new.
Yeah.
Um, so another question here is, what are the primary kind of challenges you come up against when you're trying to mimic the complexity of a human body in a microfluidic system?
Well, okay. The the difficulty is what we have to we can't build a full model, right? We can't have 250 organs and etc. like that. So we have to ask which are going to be important to the question we're asking, and then we have to ask what is important in terms of the biological detail that needs to be captured in these systems. So part of it is, you like Einstein said, you know, a model should be as simple as it needs to be, but no simpler. And so part of it just depends on, what is the question you're going to ask, and what do you need to capture in order to answer that question well. So if it's, 3 cells, it's fine. If it's you need 15 cells organized a certain way, that's much more difficult to do. Doesn't mean you can't do it, but it becomes much more difficult. So in terms of these kinds of systems, we don't build models with more than 5 organs. 5 organs, but we could. Uh, and actually, in my own lab, we built one model which had 14 compartments, I think 11 organs. So you can physically build it, but it's cumbersome, and in terms of the information you get, it's you have to ask about whether it's going to answer the question you want. But we have built up to a system with 14 compartments, so it is possible to build more. But basically, in terms of like Hespros, the company, we haven't found any real reason to go beyond 5 compartments.
No,
And
that
again,
makes sense.
depends on the question.
No, fantastic. Lots of questions have kind of come in about how you measure and maintain the long-term functionality of organ models on a chip, especially when you're testing drug responses.
Yeah. Well, it is always an issue. Um, we typically are fairly confident going out about 30 days in most of our models. That's based on law of experience. You know, there's some models when we first started with, getting 7 days was good. Um, Part of it is understanding the medium and the interaction of the medium with the cells. Sometimes it is how can we remove buildup of inhibitory metabolites. Um, a lot of it has to do with how do the cells interact with the surface chemistry. Now, at Hespros, now at Cornell we use serum sometimes, but at Hespros, all the media is serum free. And the reason serum free is that gives us very precise control, because if we have serum in there, there are things that we don't know what they're doing. And they may be very, very helpful, but they can also be inhibitory. So, so we try to build a serum-free media for most of these systems. And again, that takes a fair amount of expertise to do that well. Um, but again, most of the time we can run systems 30 days. 30 days is kind of the magic number from the way we think about things. Because we look at normal drug development, a lot of times a 30-day trial is kind of the standard. We have run systems, so you can run systems longer than that. Particular system maybe out 70 days or so. Uh, and some systems are, at least in my lab, have been more difficult to run. I can do a blood-brain barrier out maybe 2 weeks, but not much farther than that.
hmm
So there are some which are just more complex, but a lot of it is you have to work with the biology. A lot of it can be a fair amount of experimentation to get it, and it's not always easy. Now, with the company, we offer I forget how many organs, say 12 different organ compartments.
hmm
So we're fairly confident those ones that we can operate on a long time. If you gave us a new organ compartment to build, it might take a longer time to be confident we could build it on out.
hmm
But we um, you need to understand the nutrition of the cells. Uh, Also, cells once they're removed from the body will tend to start to drift genetically, and
Yes.
that
hmm
makes much more difficult too. So you want to retain that genetic part. So it's uh, that's probably the biggest challenge actually in making a new system where it's an organ that we have not worked with before.
Yeah.
So can we do that? Uh,
Hmm.
and sometimes it's uh, sometimes you just need the physical property, like a fat. Maybe we don't need the metabolic property. We just need fat in terms of absorption and stuff like that. So sometimes you don't need to build a very precise model, and fat could live a long time, etc. But if I want all the metabolism of fat, then that becomes a much more difficult problem. So if I'm
Mm.
worried about just using fat to measure partition of compounds in the blood from the bloodstream to fat, then it's not so much a challenge. But otherwise, it could be much more of a challenge.
Yeah.
Uh,
No, fantastic. So let's have a little look here. Someone has asked if it's possible to retrieve cells and tissues from body on a chip and do downstream molecular assays like flow cytometry and qPCR come into your processes?
yes, uh, we will routinely recover cells, and we often will do like post-mortem analysis on the cells to learn a little bit
Hmm.
more, and as you would if you're doing an animal study. Basically the same thing we do with an animal, you can do with these systems. Um, the uh, the systems you can are made put together. You can take them apart. You can remove the cells if you need to. Uh, the only thing is that's pretty much an input. You don't put them back together again. Uh, Now it is possible though to, for example, use like a biopsy needle and remove just a small amount of cells if you
Hmm.
wanted to look at them separately while the process is still running. You can do that as long as you're not taking. If I have a small organ system there and you're removing 50% of the cells, that's not the goal. But you could probably remove 5% of the cells and still have the system functioning reasonably well. It depends on what you want, but taking the system down at the end and looking at each of the organs would be something we could do routinely. And we do use a lot of microscopy and other things like that um, to look at the system. And again, the omic technologies you could use also on uh, if you want to look at some of the things from inside the cell at least.
Hmm. No, fantastic. Thank you
So
ever so
it
much.
is possible to do a kind of like a biopsy if you needed
Hmm.
to, but you're not working with a large organ, so it has to be you have to be careful about how you do it.
Yeah, I can imagine. Great question here, actually, that's asking, do you anticipate that these systems will be accessible and affordable to university research labs, for example, at some point soon?
Okay, so the answer is yes, although let me give you some qualifications on that. So we don't sell systems.
Hmm.
Okay, so that would be, uh, if somebody had something they wanted to do with Hesperos, they could work as partners and we could put a grant in for that, etc. So, I mean, it could be done that way. And there are cases where we work with other university researchers on grants and things like that. So that would be a possibility. Um, there are other people that make MPS systems that sell those systems. And again, it's, you know, the question then becomes, do you have the in-house expertise to make good use of the systems or not? And a lot of the systems I think are fairly tricky, and so that it's, you know, if you've done this, 100 times, then it's not such an issue. But if you haven't done it before, then it becomes much harder. So it's certainly possible. I think, like if you want to use our systems, it would be you'd want to work with the company on a problem. So it'd have to be something we were interested in, you were interested in, and we could get a government agency that's funding it to be interested. Um, but again, like I say, other people sell devices, so you can try using that. That would depend on the device and the question you want to ask.
Hmm.
You could also try to build your own systems, but I think that is That's a bit tricky. Yeah, uh, when we most all the times, many of the cases we use, the company approaches this. We're making a new system fit their problem,
Hmm.
not taking an existing system and necessarily. Sometimes we can, uh, but a lot of times the question is asked in such a way that we need to add something to the system to it. Now, sometimes if it's you know heart liver problem they have, that's kind of a standard system for us. So we can do that. Or if it's just they want to know what's happening in the neuromuscular junction, we can do that. But if it's a system which is question different, that requires different organs at different sizes, etc. Um, we may have to fabricate a whole new system, and we just build that into the cost. We say, if they come to us with an idea, we say, this is what it would cost to do those experiments. If we have to build a new system to do it, then we build that into our cost.
No, that makes a lot of sense. So let's have a look at some of the questions that have come in. Um, this one says, can your system be used for early discovery of unknown compounds?
Okay. I'm not quite sure I get unknown compounds, but
Hmm.
let's see. Um, the unknown compound, I would have to have some reason. So if I was looking for a compound that did something,
Hmm.
I would have to know what it did to fish it out of the system. If I have a way of fishing it out of the system, I could do it. But if it's an unknown compound, which I don't know what it does, I can't do much. Uh, so I don't know if that may not be clearly what the question was, but that's.
No, that's no problem at all. Let's have a look, see if there's a good the question that we've got in here. Um, let's have a little look. Someone here, I think this could be a really nice way to kind of close off the session. Um, they said you may have mentioned it before, but as we have people joining at all points in the Ask Me Anything, I think it's a nice way to kind of close off the session. What inspired you to start working on the body on a chip technology?
Well, in my case, in part was because of my daughter Down syndrome. Uh, the other part was I'd done a lot of mathematical modeling of cells and stuff in biology. It didn't always have the impact I thought it would. Uh, so I was looking for something where our modeling expertise would have impact. It was clear PBPKs were having impact already. So the question was, with the PBPK, how can I make that better? And so the idea was, if I have a PBPK and I also have the cell culture analog, I can make the PBPK much better and I can be much more confident results. So part of it was driven by, something maybe to help people with, special medical issues. But the other part was also, I think, the idea that we could use our skills in modeling combined with skills in cell culture to build systems which give us answers to questions that no one was looking at before. And again, we thought that uh, it's fairly clear that animal models are not always a very good use of resources. So having a human surrogate could be very useful too. So it was kind of a combination of those things that I think inspired us to try to look at the idea.
Hmm. No, fantastic. Well, I think that's actually a lovely way to close off the session. So thank you ever so much for joining us today. Unfortunately, that does bring it to a close, but it's been an absolute pleasure speaking with you. From the look of the comments, everyone's loved it as well. So yeah, thank you ever so much.
Okay, well, I appreciate the chance to do this. I've enjoyed it.
Wonderful. Thank you ever so much. And then to everyone who tuned in, thank you so much for being here and for your fantastic questions. Before you leave, don't forget to download your certificate of attendance if you haven't already. We'd love to see it if you share it on LinkedIn. So be sure to tag us and we can engage with your posts and hear your key takeaways from today. If you enjoyed this session but dropped in late, don't worry, you haven't missed anything because the full AMA will soon be made available on demand. And feel free to share the link with friends, family, or colleagues who you know might be interested. And if you want to hear more from Professor Schuler, I am delighted to let you know that he'll also be presenting at the Innovations in Disease Modelling online symposium on the 23rd and 24th of April this year. So you can check out the link in the handout section to see more. And once again, thank you everyone for your time and participation. And we look forward to seeing you at the next Ask Me Anything in February, which will be focused on epigenetic biomarkers for early disease detection. See you later.
Thank you.